Yeah, my name is Yunji Tong. I'm from Purdue University. If you do not know where it is, it's 2 hours from Chicago. And my talk is about new discovery using fMRI to study sickle cell disease. Okay, so I'm very appreciate Megan to talk about basics about sickle cell. I'm not going to repeat it. So I just want to say that among all the MRI technologies, people use T1 with the image, people use T2 flare to study sickle cell. However, very limited study people using fMRI to study sickle cell. Among those studies, actually, most studies study resting state connectivity, right? So in this talk, actually, what I want to do in our studies, we try to use fMRI to explore the physiology part of the sickle cell to try to see how the cerebral blood flow or oxygen level in the sickle cell only use fMRI data. So before that, I would like to give a little bit of background of my my group and my study. And actually, I've been interested in using fMRI to study physiology for quite some years, and we developed several methods try to understand, especially about cerebral blood circulation using fMRI. So in about 3-4 years ago, we started interested in the Bose signal in the large blood vessels, right? For example, in the internal carotid artery, in the suprasacral sinus, and of course in the global mean. So this is study we try to do is, in this study we try to extract temporal series from these large blood vessels, and we try to do cross correlation between them. And using the cross correlation, we try to map out how the cerebral blood flows through the brain, right? So the idea is very simple. We work on the resting state fMRI data. So basically from T1 and T2 with the image, we can extract very accurate, internal carotid artery as shown down there. And then we can also extract, suprasacral sinus very accurately. And so basically what we did next is we project these masks on the resting state data. Then we can extract corresponding both fMRI time series from these corresponding structures, right? So here is a result I want to show you. This is from resting state data from healthy subjects. And at the top left figure, you can see I'm plotted red is the both internal carotid artery signal reversed. And the green one is the global mean signal. And on the lower left one, I'm showing you the suprasacral sinus signal which is in the blue, and also the same global mean signal, which is at background as gray. And you can clearly see, first of all, these three signals, they have very high correlation, right? So again, correlation value is about 0.7. It's very high correlation. And secondly, as I mentioned, the internal carotid artery signal is reversed, is anti-correlated with the global mean signal and also anti-correlated with the suprasacral sinus signal. And however, the global mean signal and the suprasacral sinus signal, they're positively correlated, right? You can see at the bottom left. And last thing is the most important thing is if you calculate the delay between them, you can clearly see that the internal carotid artery signal is about 2.9 seconds earlier than the global mean signal, which is about 3.6 seconds earlier than the suprasacral sinus signal. Okay, so if you add these things up, that you can see that from both fMRI, if starting from internal carotid artery, it takes about 6.5 seconds for it to reach the suprasacral sinus, which is roughly similar to the literature, right? That's what we call the cerebral transit time. Okay, so actually we know exactly why the internal carotid artery signal is anti-correlated with the suprasacral sinus signal, right? Because this is due to the susceptibility effect. Because we know at different blood vessels, they have different content of oxyhemoglobin and deoxyhemoglobin. And based on the content difference, you either have a positive blood signal, or you have a negative blood signal. So based on a very simple model, we can know that on the top left, we can calculate this kind of what we call threshold, which is about 86% oxygen saturation. So if the blood vessel, right, where you get your blood signal from is lower than 86%, which is the suprasacral sinus, you stay on one side of the curve. That means your signal is positive. And if you're a blood vessel, in the blood vessel, you have higher oxygenation, which is beyond 86%, you come like internal cardiac artery, you become negative, right? So that's clearly explained that the both signal from the internal cardiac artery and the both signal from supersatural sinus, they're anti-coordinated. How about the global mean? And we all know that both signal is very, very sensitive to the venous and veins, right? So when you calculate the global mean, the signal is very sensitive to the venous in the brain and the vein in the brain. So in that case, the majority of the venous and vein in the brain, they are below 86% oxygen saturation. So that's why when you calculate the global mean, the average, the lower left, you can see the global mean signal, most of them are on the 86, one side of 86%, the same side as supersatural sinus. That's why the correlation between supersatural sinus and global mean, they're positive. Okay. So now we're back to the sickle cell study. So this study actually we borrowed resonance data from my data from existing study which is still going on, and our collaborators are Dr. Wang and Dr. O'Brien from Indiana University School of Medicine. So far we have 23 sickle cell patients, and we have 10 age gender matching non-sickle cell controls who are African Americans. Okay, so here, because this is an existing study where we're not able to extract internal cardiac artery because the scan was not deep enough. So we say, why don't we just extract supersatural sinus from the resting state data, and why don't we just compare that with the global mean signal? The hypothesis was that for the sickle cell patient, their cerebral blood flow is faster. So what we say is when you do the cross correlation between the global mean signal and the supersatural sinus signal, when we calculate the delay is supposed to be shorter than the healthy subjects. That was the hypothesis. Okay, so that's what's shown there. So this is the result from all the subjects I mentioned. So here on the left, you can see I plotted every individual subject, healthy subject, sickle cell subject. I plot their maximum cross correlation value corresponding to their delays, right? So here you can see on the lower right corner, that's the where you find the most healthy subjects. So these healthy subjects, they behave exactly like what we predicted. They have a positive correlation values, very high. That means the supersatural sinus and the global mean are positive correlated. And delay time is about 2 to 4 seconds. That means the blood arrives at the global mean at brain first, then later they reach the supersatural sinus. And the interesting thing is about 11 out of 23 sickle cell patients, they behave exactly like the healthy subjects. Like you can see the blue dots on the lower right. However, the shocking thing is that the 12 remaining sickle cell patients, they behave totally differently. So the correlation between their global mean signal and a supersatural sinus signal are reversed. And it's pretty significant. It's minus 0.6, minus 0.7. And interestingly, their delay is reversed too. That means for those sickle cell patients, it seems like the blood, arrive at the sickle and the supersatural sinus before they travel through the brain, right? So that's kind of puzzling. But before we jump too deep, let's talk about MCCC first. I'll talk about the maximum cross correlation coefficient first. Why it's negative? Okay, I jump ahead of myself. So let's first talk about why these 12 remaining sickle cell subjects, why they are separate from the rest of the sickle cells subject who behave exactly like healthy subject, right? So what are those 12, interesting sickle cell patients? What make them different? So here, what I did is I plot for all the sickle cell patients, their blood measures in terms of hemoglobin level, in terms of the hematocrit, and in terms of the sickle hemoglobin, right? So you can see I plot their blood measures versus their MCCC value, right? You can clearly see that for the sickle cell patient who have a lower hemoglobin level, who have a lower hematocrit level, and who have a higher sickle hemoglobin, they tend to be the one who have a negative MCCCs. So that means, in other words, right, that the sickle cell patient who suffers severe symptoms, who tend to be the one who give you negative correlation between global mean and supersatural sinus. So why that's the case? So we have the, after reading Mann's paper, we had several hypotheses. One of them is that we think this is due to what we call the vascular shunting. So basically what happened is actually the blood from your internal carotid artery, actually, because of shunting, they, populated, they flow faster and they bypass some, capillaries. So in that way, so they will make the blood in the venules and veins are highly oxygenated, not as the normal subjects. So basically, this is the model we try to explain this. So as you have as your sickle cell symptom gets severe, you have a shunting happens more and more. So that pushed you many, brain voxels become highly oxygenated. So that push them pass over the threshold. So that in that way, when you calculate the global mean signal actually is anticardiac with your supracyclic sinus signal. How many minutes I have? 10 more minutes? Okay. So then I probably gonna jump this. If you have time, I'll come back to this. So okay, so that if that's the reason, then let's come back to these figure again. So we demonstrate that it's 5 more minutes. So is it possible that a maximum cross correlation coefficient can be used to as an imaging biomarker to tell if the sickle cell patient is severe or not. However, we haven't addressed the big elephant in the room, which is why the delay is totally reversed. That means for those special sickle cell patients, why it seems like their blood arrived at their supracyclic sinus before they travel through the brain. What's the reason? We haven't found the reason until a couple of days ago, which I know I have to come here to give a talk. I squeezed my students so hard. So we came up with something that I think is making a lot of sense, but I would like to listen to the experts, listen to your opinion. So basically what will happen is really also due to vascular shunting. So this is what happened. So before going there, I want to introduce you correlation coefficient calculation for a severe sickle cell patient. So here you can see on the top corner, I'm calculating the correlation coefficient, right, for between the global mean signal and the supracyclic sinus signal. And you can see that one that only is the maximum correlation coefficient value. That's the red dots. That give me the negative correlation, right? That give me the negative correlation. That means the global mean and the supracyclic sinus is negative correlated. And that give me the 3.3 seconds delay. That means the blood arrive at the supracyclic sinus earlier than it arrive at the brain. However, there's another competing peak, which is not very far from that peak. If you go to the second highest sickle cell, second highest cerebral correlation coefficient value, which is 0.5, is still pretty strong. And in this case, the delay is minus 3.9 seconds. So what does that mean is there's a competing effect that there's a second part of the brain, a second part of the blood. Actually, they flow exactly like in regular blood. They go through your brain first, then later on, about 3.9 seconds later, they arrive at your supracyclic sinus. So based on this figure, we understand there might be 2 different arrival time for your blood. So this gives us a new model. So basically for the blood, after they reach your brain, right, one part of your blood will go through this, what we call arterial venous shunting. So they will bypass most of the capillary and reach your supracyclic sinus early. And this blood actually carries high oxygen level. While the other part of the blood will go through, the harder path, which is the capillary bed. And the second part of the blood actually traveled the regular way. So that will arrive your supracyclic sinus later with the typical oxygen saturation. So by doing this, we give, we use these to try to explain both the negative MCCC and also the 2 different delays. Okay, so here's the conclusion. For the time of sake, I'm not going to read of this. And as I said, these findings are initial and also exploratory. I really welcome the audience, especially this group, to really give me a lot of feedbacks, and we can work on these patients more. And I would like to thank my group, especially those 4 people. And they did the most work, and also thank my collaborator. Thank you.